Welcome to part 4 of this online training program on adaptive designs and clinical trial simulation. In this module, in this video, we will continue the discussion of adaptive proof of concept or dose-finding trials that support dynamic response adaptive randomization. I will begin by quickly going over the key resources that we have prepared for this training program. This includes, of course, the R package called Mediana Designer that I mentioned multiple times before. It's a free open source software tool that we have built to enable all of you take advantage of efficient simulation-based approaches to clinical trial design. And this is the package that will be featured in this online training program. You can download and install it using the CRAN website. This is the first link that you see on this slide. In addition to that, we've prepared numerous other resources, including detailed description as well of the underlying statistical methodology, as well as technical manuals. They could be found on our website at mediana.us. Again, you can follow the link shown on this slide. And we've also prepared a detailed online manual with multiple case studies to help you come up to speed with this software tool. You can find it on the GitHub website. You can follow the link shown at the bottom of the slide. I would like to mention that the software tools that we will be using in this training program are available using two deployment models, if you will. You can either download the Mediana Designer package, as I explained a minute ago. You can then install it on your computer and run the R code that you can either download from the package's online manual, or of course you could write your own code, or alternatively you could take advantage of web applications that we have built. These applications are available on the cloud platform we have set up, and this slide shows you how you can access those applications. You can see the login information as well as the password, and there will be a separate web application for each function that I'm going to introduce in this online training program. For example, there will be a separate application that supports simulation-based design of response adaptive trials that are dose finding or proof of concept in nature. And what is great about these web applications is that you will have access to the same functionality, which means that you can run simulations to assess operating characteristics of the same classes of adaptive designs using these online tools without having to install any software on your computer. But in this training program, which includes this module, this video, we will focus primarily on the option of running the software on your computer, and this will be done later in this module. So the main goal of this video is to introduce software tools for phase 2 trials that support response adaptive randomization. Before we get into technical details, as I said earlier, software for trial simulation plays a key role in this online training program, and that's why we've built a large number of free software tools to help you and other research teams around the world design clinical trials with adaptive, data-driven decision rules. And the reason we pay so much attention to clinical trial simulation is that careful, disciplined planning and the comprehensive simulation-based assessment of key design characteristics under potentially multiple scenarios, making different sets of assumptions about the magnitude of the treatment effect or about the individual design parameters, would ultimately provide a solid foundation for a robust adaptive trial. The Mediana Designer package includes multiple functions, and in this video I will introduce the AD-RAND function, and I'll also mention the corresponding web application. AD-RAND stands for Adaptive Design Randomization, so this is a direct reference to the class of adaptive designs that it supports. It supports designs with response adaptive randomization. In addition, when you install the package, you can launch a web application. You can do it by calling the ADRAND app function shown on this slide. And by launching this application, you will have access to a user-friendly graphical user interface. We call it a web app, but it's actually going to be running on your computer. And this is a very helpful, once again, user-friendly tool that will help you design adaptive phase 2 trials, adaptive dose finding trials that enable this adaptive feature, that enable dynamic response adaptive randomization. And for more information about the underlying statistical methodology with relevant examples, you can check out the technical manual. The link is shown at the bottom of the slide. I'm going to illustrate simulation-based approaches to designing phase 2 or dose finding trials with adaptive randomization using this case study. We call it case study F1. That's how it is labeled in Mediana Designer's documentation. I'm going to introduce this case study in the next section. This is a high-level description of key features of the phase 2 dose finding trial that will be used in this case study. We went over this clinical trial in part 3, so we'll provide a very quick description. You can see here that this is a phase 2 trial in patients with partially responsive major depressive disorder. There are multiple trial arms. It's a dose finding trial. We'll have a placebo arm. In addition to that, there's an experimental treatment that will be administered to the patients at 4 different dose levels. They're defined on the slide. This is a 100 milligrams per day, 200 milligrams per day, 400, and finally 800 milligrams per day dose. The primary endpoint in this trial is a continuous, normally distributed endpoint that is defined as the change from baseline to the end of the treatment period, which is going to happen at 4 weeks. So it's a change from baseline to 4 weeks in this total score, which is known as the MADRS total score, which stands for the Montgomery-Åsberg Depression Rating Scale. A multi-stage design will be utilized in this case study to support this data-driven option to update the randomization schedule. 4 stages will be used, and 50 patients will be enrolled in this stage. As far as the response adaptive randomization approach is concerned, as we discussed this in detail back in part 3, patients in stage 1 will be equally randomized to the 5 trial arms, and then patients in the subsequent stages will be randomized to the trial arms using a response adaptive randomization approach, which means that all of the information, the cumulative information on the dose-response relationship for the primary efficacy endpoint, will be utilized at the end of each stage to make decisions about how to more or less optimally update the randomization scheme for the next stage. And the goal here would be to utilize a Bayesian approach. That's why we see here a reference to posterior probabilities, and we would then like to assign most patients to the most effective doses. We're now ready to begin discussing the code that will need to be written, the specifications, the required parameters for the ADRAND function that enables simulation-based assessments of the proposed response adaptive trial design for this trial, in patients with major depressive disorder. And the R code that would be required here is shown on the next several slides. At the very beginning, this is a requirement for all of the functions included in the Mediana Designer package, is that we first need to initiate a list of parameters. And then the individual parameters would then be placed as individual elements on this list of parameters. The first parameter that we're going to pass on to this function is the specification of the endpoint type. As you can see on the slide, the endpoint type here is set to normal, So we assume the primary efficacy endpoint to be continuous and normally distributed. Secondly, we specify the direction of favorable effect for the primary endpoint. In this case, this is a key feature of the MADRS total score that will be used for in the primary efficacy analysis, is that a larger reduction in this total score would indicate a beneficial effect. And therefore, the direction parameter in this case will be set to lower. If this is not the case, and if it's perhaps in some other clinical trial, a higher value of the primary efficacy endpoint is associated with a beneficial effect. In that case, this direction parameter will need to be set to the keyword upper. And the last parameter shown at the bottom of the slide defines the trial design. And subsequently, we'll also talk about how to specify the treatment effect assumptions. The most important feature of this design would be the dose levels for the experimental therapy that will be administered to the patients in this trial. And the specification is shown as follows. The dose levels vector is set to the values 0, 100, 200, 400, and 800. The zero value here, of course, corresponds to the placebo arm, and the numeric values show the dosage. In this case, the dosage would be in milligrams per day for each dose level of the experimental treatment. The next set of specifications here deals with the treatment effect assumptions across the dose range in this clinical trial. And as you can see here, we are specifying separately the means and the standard deviations, and that is because, of course, the primary efficacy endpoint is expected to follow a normal distribution. For the control arm, we only need a single value for the mean and a single value for the standard deviation, and these results here reflect historical data available to the trial sponsor. And here, once again, I'd like to point out that we expect to see a reduction, and therefore the control mean is equal to -10. So patients naturally, as they progress, would force the MADRS total score to go down. At the bottom of the slide, we see a similar set of specifications for the means and standard deviations across the 4 dose levels for the experimental treatment. And therefore, when we see the mean specification, we will need to provide a vector of 4 values, and they are given by -12 all the way up to -18. We can see here that as the dose increases, we expect a larger reduction in the primary efficacy endpoint. And as far as the standard deviations are concerned, a common standard deviation of 14, which is the same as the standard deviation in the placebo arm, is assumed across the dose range in this clinical trial. As I said a few minutes ago, this will be a 4-stage design, and the first specification on this slide gives us an option to explicitly define the number of enrolled patients in each trial stage. In this case, since we have 4 trial stages, a vector of 4 values will be passed to this stage sample size parameter. For the AD rand function, each element of this vector is 50. So we expect to enroll 50 patients in each trial stage, of course, across the 5 trial arms. The next parameter here, which is denoted by delta, defines the clinically relevant improvement over placebo. This is the parameter that we referred to several times back in part 3, where we talked about the underlying statistical methodology. And at that point, I pointed out to you that the way the randomization scheme is updated for each next stage in a clinical trial is that we model the dose-response relationship in the trial, and we compute appropriately defined posterior probabilities of achieving target efficacy. So how do we define target efficacy in the context of this case study? Well, it is defined using this threshold. So the target efficacy profile would then be defined by a clinically relevant improvement over placebo, and therefore ultimately the randomization scheme for each next stage in the trial would be driven by the posterior probabilities of achieving a 6-point improvement over placebo in each individual dosing arm in this trial. And when we talk about adaptive dynamic response, adaptive randomization, there is a balance parameter. We denoted it by rho back in part 3. In this case, this balance parameter is set to 2, and this parameter defines or controls the degree of balance for patient allocation for the next cohort. As we said when we talked about the underlying statistical methodology, with a smaller value of this parameter, the patient allocation across the dose range, across the dosing arms, is more likely to be balanced. And when we choose a larger value of this parameter, for example 2, this would be an example of a larger value, then the allocation will be increasingly imbalanced. We also need to specify a few parameters that define the patient enrollment and patient dropout processes. In this case, we would assume that patient enrollment period would last for 36 weeks. In this case, and in general, when we look at all other functions included in the Mediana Designer package, I would like to point out that the time unit is not explicitly specified. What's important to remember here is that the same time unit needs to be used across all of the function parameters, across all of the specifications. In this case, the time unit would be weeks. That would be how we measure the length of the treatment period in this case study. That's a 4-week treatment period. Likewise, when we define the length of the patient enrollment period, this will be a 36-week enrollment period. The median enrollment time will be set to 24 weeks. The median enrollment time is a parameter that specifies the milestone by which 50% of the patients will be enrolled. So in this case, as you can see, we're not making a basic assumption of uniform patient enrollment. This is not a realistic assumption, as I've said before, and I'll continue to this point. When we set up clinical trial simulations, it will be important for us to mimic key features of patient enrollment and patient dropout processes as closely as possible. In this case, this particular specification states that half of the patients, 50% of the patients, will be enrolled during the first 24 weeks in the trial, so it will be a fairly slow enrollment process, but then it's going to pick up, and in the remaining 12 weeks of the patient enrollment period, the other half of the patients will be enrolled. Then, as I said a minute ago, the length of the treatment period needs to be specified. It will be 4 weeks. And finally, towards the bottom of the slide, the patient dropout rate is defined. We expect 5% of the patients to be lost to follow-up, to drop out of the trial by the end of the treatment period, which will be at the 4-week time point. The next set of specifications is somewhat technical. I will go over it fairly quickly, and it deals with the model averaging approach used in the MCP-Mod method. That is the method that is used in this function for providing the best possible description of the dose-response relationship for the primary efficacy endpoint. And this method requires that we pre-specify a set of dose-response functions that describe potential plausible shapes of the dose-response relationship. Those functions would need to be pre-specified. We also need to pre-specify parameters that explicitly define the shapes of those functions. In this particular case, 4 different dose-response functions will be specified. A linear function that does not require an explicit specification. We're also going to assume an exponential dose-response relationship described by an exponential model, and that the shape of that dose-response model is quantified using the delta parameter. In this case, this delta parameter is set to 400. The next model to be used as part of this MCPMod method is the Emax model. And the parameter that needs to be specified for this model is the Emax, is the ED50 parameter. This is the dose at which half of the maximum effect is observed. And here we assume that this will happen at the 600 milligram per day dose. And finally, the last model to be included in this specification is a logistic model. And this one has 4 different parameters. There are 2 linear parameters and 2 nonlinear parameters. So those nonlinear parameters need to be explicitly specified here. They are the ED50 and delta parameter. They're set to 400. That's again the dose where half of the effect is expected to be realized, and then delta is set to 50, which is yet another parameter that describes the shape of this complex logistic dose response model. Once again, I'm not going too much into detail here. I would like to, if you're interested in finding out more about this particular specification, I would like to encourage you to download the technical manual as well as relevant resources that provide information about this powerful methodology, the MCPMod methodology. And we're almost done. There are only 2 more parameters that need to be specified. The first one is the one-sided type 1 error rate, or the alpha. It is going to be set here to 0.025 or 2.5%, which obviously corresponds to a 2-sided 5% significance level. We're going to set the number of simulations, because we're going to here rely, of course, on simulations to assess key operating characteristics of the proposed adaptive design. We will going to run 1000 simulations. Now we're done with all of the specifications. All of the required parameters have been given, have been specified, and the resulting list of parameters is going to be now passed to the AD-RAND function. This function is going to output an object that we're going to assign to results, and then this object includes all of the information about the assumptions that went into the simulation process, as well as the key simulation characteristics. And then to be able to produce a detailed, really comprehensive simulation report that you can easily then save to your computer, and you can use this for future reference, you will need to take this results object and you need to pass it to the generate report function, and then specify the file name for this function. The function is going to generate a report. It's a multi-page document in a Microsoft Word format, and it's going to save it to your hard drive under the name case study f1.docx. So we're all done with this very quick review of the code that would be required to be able to run clinical trial simulations for this case study. And I would like to point out to you that this R code that I've just walked you through, as well as the simulation report, can be easily downloaded from Mediana's site on GitHub, and this is the link that you can follow. So as I said, you can download and review the simulation report generated by the AD-RAND function for this case study. This report presents the design's operating characteristics, and here I will highlight key points related to the use of dynamic response adaptive randomization in this phase 2 study. First of all, very quickly, I would like to show you the title page of the simulation report. Once again, it's a Word document. It provides a detailed summary of all of the assumptions that went into the process of designing this trial. The assumptions are presented in tables 1 through 7. On this screenshot, you only see table 1. Again, this is a multi-page document. And there's also, by the way, a figure that plots the assumed shapes of the dose-response relationships that were used in the MCPMod method. And the simulation results, the most important operating characteristics, are summarized in the last 3 tables included in this report. Those are tables 8, 9, and 10, and I will present those tables on the next 3 slides. We're going to begin with the summary with table 8. It shows us the average sample size by trial stage. We can see here clearly unequal sample sizes across the trial stages, and specifically when we look at the mean number of patients that were used in stage 1, that's the number of 69.6, very close to 70 pages. And at the very bottom of this table, we see that only 13 patients contributed to stage 4. Why is that? Well, this is due to the pipeline effect that I described back in part 3. And to quickly remind you, pipeline patients are those patients who are enrolled in a multi-stage adaptive trial between the end of the current stage, where the randomization scheme should be updated, and then there's a bit of a gap, there's a bit of a delay. We'll have to wait until the actual time point where the randomization scheme is updated. And as a result, instead of switching to the new randomization scheme for the new stage, those pipeline patients actually follow the old randomization scheme from the current stage. And when there are too many pipeline patients, a trial design with adaptive randomization will be less effective. Back in part 3, we call it a blunting effect. And the main reason we see so many patients here in stage 1, which is again almost 70 patients on average instead of 50 patients, which is the expected sample size for this stage, is because 20 of those patients are pipeline patients. They should have switched to the new randomization scheme for stage 2, but they were randomized to the trial arms before the information was available to update the randomization scheme. Therefore, they had to follow the old original randomization scheme for stage 1, which was equal randomization. So, of course, it's natural to ask, how do we address this issue? One could consider reducing the size of the first stage and instead increasing the size of the later stages in this trial. And then you would run additional simulations, another batch of simulations, for example, to assess the impact of those changes on the operating characteristics of this design. And as we've said before, simulation-based design of trials with adaptive rules is generally an iterative process. And that's the reason that I would like to encourage you to take advantage of this software tool, this R package. In addition, other tools could be used. And the ultimate goal here would be to go through this iterative process and ultimately arrive at a design that works best for a particular application, for a particular trial. The next table, table 9, also presents a summary of sample sizes, but it does it from a it looks at the data from a slightly different perspective. This particular table shows the mean number of patients in each of the 5 trial arms. We can see here that by the end of the trial, when we're done with the last stage, we expect the following mean sample sizes: 38 patients on average in the placebo arm. And then as we go through the 4 doses of the experimental treatment, we notice that the mean sample size starts at about 35 at the lowest dose. And we now go down to the bottom row in this table, we see that the mean sample size is expected to go up to over 40 patients, specifically 41.5 patients at the highest dose. And why does this happen? Well, this happens because here we have implemented an adaptive design that supports adaptive data-driven updates to the randomization schedule. And this table shows then the main conclusion here is that it is beneficial to apply adaptive randomization rules. Even though we said a minute ago that the adaptive design was not applied in the most efficient way due to the pipeline effect, but still, based on the predefined rules for updating the randomization scheme, we can see here that the dose response data were analyzed at the end of each stage, and then the average dose response model was derived based on the MCP-Mod method, and it was used to compute the posterior probabilities of achieving target efficacy for each individual dose compared to placebo, and that those posterior probabilities ultimately helped the trial sponsor update the randomization scheme for the next trial stage. So this table shows us the adaptive randomization approach is expected to favor the more effective doses, and these are doses 3 and 4, and there's clearly a lower probability of assigning patients to the less effective doses, and these are doses 1 and 2. And this is once again the main benefit of using an adaptive randomization approach in a dose-finding trial. The last table of the simulation report, table 10, presents a comparison of the traditional design without adaptive randomization. It relies on a fixed randomization schedule to the proposed adaptive design. We can see here that the probability of success, overall power for the two designs is quite comparable, 77.2% for the traditional approach versus 76.2% for the adaptive approach. And here, by the way we define power is quite is how power is commonly defined in those finding studies. We rely here on the probability of a significant dose-response relationship, and that probability is assessed using the model averaging approach derived from the MCP-Mod method. And the main conclusion here is that the adaptive design provides the same probability of success in the trial compared to the traditional design, but at the same time, it achieves a very important goal of assigning more patients to the more effective doses. We are ready to wrap it up for this video. In parts 3 and 4, I would like to remind you that we have gone over key design as well as statistical considerations, operational considerations for adaptive dose-finding trials or phase 2 trials that utilize response adaptive randomization. We've also went over case studies as well as an efficient software tool, the Mediana Designer package, that you could use to support efficient clinical trial simulation for this class of adaptive designs. And the ultimate goal here would be to help you design adaptive trials of this kind if you're interested in exploring response adaptive randomization in your own trials. I would like to thank you. I appreciate your interest in this online training program. I would like to encourage you to watch the other videos and share information on this training program with your colleagues.